Rule 1 answers the question, how does the outside of an organism, the environment, change life's inside? When you have something that grows up in a different environment, how does that environment shape that organism? And this is kind of an abstract question, so I want to make it a more specific. More specifically, what's wrong with the DNA in Mono Lake? I'll explain that. But the rule that answers the question is that the chemistry of the inside is constant, because each element has a job that comes from its place on the periodic table. Now, as we begin, I have periodic tables for all of you. So, if you could just pass that back. And let me make sure that I have enough here. So everyone can get a periodic table. And you can refer to this. This is sort of your roadmap as we go through. So DNA's sequence will definitely change. A lot of things about the inside of the organism, the information inside the organism, may change. But its chemistry does not change. I don't know if you've ever been to Mono Lake. Honestly, to tell you the truth, I haven't. But I've seen pictures, and I've seen paintings like this. It looks like a really nice place. It's a big lake on the California-Nevada border. And it's a very unique environment. And some scientists, about 2010, some scientists had a big press conference, and they announced that they had found arsenic-based DNA in Mono Lake. A lot of people said, that's really cool. A lot of people said, how can that happen? Well, the reason why they're looking in Mono Lake is they're looking for weird life in a weird environment. And Mono Lake even looks weird. Mono Lake has these tufa towers that come from all the minerals that dissolve in it from the mountains around it. It has carbonate minerals that make these towers. It has a lot of the other rocks that are around it, and that includes a lot of arsenic. And so this might look pretty, but you don't want to drink it. Now, the reason why an arsenic-rich environment is a good place to look for weird life is because the very heart of life is DNA. And the very backbone of DNA is made of phosphate, which is right next to arsenic on the periodic table. So all known DNA has a backbone that looks like this, has these phosphates. If you know a little bit about biology, the DNA makes RNA, and the RNA makes proteins. The proteins fold up, and the proteins assemble together with a lot of other stuff. And if you want to know more about that, then I can suggest Biochemistry 1 next fall. They all come together to make a cell. And DNA is at the very beginning of that. And you notice all that phosphate in DNA. Okay, well, if you look at the periodic table, the next best thing to phosphate might be arsenic. So if you look at it right there, you can see that all the columns in the periodic table, one of the things that it tells us if something is in the same column is that its properties are similar. It's chemically similar. And if you look up and down, from phosphate to arsenic, well, arsenic's right next door. So maybe arsenic could work. It's reasonable. And the microbiologists were saying, this means that the microbes in Mono Lake could build arsenate DNA. And here, look at our data. We show data that they are using the DNA to build arsenic to build DNA. I had that backwards. But the chemists had a problem with that, because chemists work with these things in solution. And any chemist who has worked with arsenic in solution knows that arsenic falls apart in solution. It's like building with straw instead of brick. Okay? In water, arsenic just attracts the water and the bonds break down. So the chemists will actually say, well, that's nice chemical reasoning using the periodic table, but we know from working with it, it won't hold together. Yes, it will. No, it won't. Yes, it will. No, it won't. Right? So we resolved this dispute in the civilized way. We ran experiments. And so we recreated the environment. We delved into the environment of Mono Lake. We recreated the environment in the lab. One of the big labs in this was right up the road in British Columbia. And the whole story of that is in the book. But you can even recreate Mono Lake yourself in your kitchen. If you have a bucket, you fill it with water, you add baking soda, table salt, Epsom salt, and borax. This is a pretty good simulation of what Mono Lake is like. And it includes these elements over here. But then, if you really want to make a good Mono Lake in a bucket version, you're going to add some of these things, including arsenic, because it has high enough levels of arsenic to be dangerous to life. The other thing is, if you leave this bucket out for a couple of months, the water will evaporate, and you'll form your own little tufa towers inside the bucket. So that's another way in which it simulates Mono Lake. This is a lot like the chemical garden that you might have seen, where you grow these sort of crystals inside a flask. This is a silicate chemical garden. So there are a bunch of experiments. There's 4 of them described in the book. A bunch of papers flew back and forth, and Science Journal sold a lot of copies. At the end of the day, I want to say, for this one story, the chemists were right. The arsenic doesn't hold together, because in a high arsenic environment, even these microbes will still keep using phosphate to hold DNA together. And that says something, because this is a low phosphate, high arsenic environment. The deck is stacked against phosphate, and yet the organism still uses it. And there are some facts about phosphate. Its chemical properties are such that it is what the organism needs to use for DNA. So if we kept doing these experiments, we would predict that if we replayed the tape and redid the experiment, that phosphate would still be used to build DNA in Mono Lake. A lot of times scientists are asking this basic question over and over again. And one of the things that they ask is, what would happen if we replayed the whole tape of life for the entire universe, not just Mono Lake? What if we rewound the thing 13.7 billion years and replayed it? Would we still be here? Or is it just kind of a random fluke that we're here? This is a big debate. One of the prominent voices that says that we would not be here, that it is just a fluke, is a biologist named Stephen Jay Gould. He wrote that the biology would be very different if we rewound the tape of life. It's very unlikely that anything like human intelligence would ever emerge again. Well, as a chemist, I look at this example of Mono Lake, and I say, is there something in chemistry that gives order to life and gives order to the universe? Something that might make it more likely that something like this would happen again. So I think that there is, and that's what the rest of this talk is about. To explain this, we have to look into life, and we have to look into what chemicals are in life and how they might have come about. That brings us to rule 2.